information about their viral microbiome. We are characterizing the bacterial and fungal and eukaryotic elements of the microbiome as well, but I come from a background of studying dengue virus evolution, so I'm particularly interested, near and dear to my heart, are the RNA viruses. So, of course, my motivation for getting involved in dengue, and I've also worked on hantaviruses and a little on influenza viruses, is the origin of emerging infectious diseases, which are invariably of animal origin, of non-human animal origin. And so in our backyard in California, we had a Yosemite outbreak of hantavirus, which humans contract from rodents, such as deer mice in that area part of the world. Of course, SARS, we picked up from probably a bat origin. West Nile virus has got many animal reservoirs. This is supposed to symbolize MERS, the Middle Eastern Respiratory Syndrome, which you can see very close human and non-human animal contact. So invariably, emerging infectious diseases have been traced to these animal reservoirs. This is an image of dengue virus in its sylvatic cycle or jungle cycle, forest cycle, where it cycles between non-human primates and canopy-dwelling tree-bulb breeding mosquitoes. And it emerged into humans from this reservoir, probably bridged by the vector. So a lot of times vectors are implicated in bridging that interaction between natural non-human animal reservoirs and humans. So an emerging infectious disease, of course, is that which hasn't been known before, or something which dramatically increases in incidence in human populations or other even agricultural or domesticated animal populations. So this is a mosquito. As I said, it's often the bridge for increasing these contact opportunities between humans and non-human animal reservoirs of potentially infectious or emerging infectious diseases. And so dengue is one very close example where we can look at the distribution of pathogens in sylvatic cycles. These are dengue viruses shown in pink from the different serotypes, and then the human forms are shown in black and white. And we can see that because all forms of dengue virus do have sylvatic cycles, it suggests that, and that they're more closely related to the viruses that circulate amongst humans, that these viruses diverged first in these animal cycles and then later independently jumped into humans. So when we broaden our look at the distribution of the viruses that dengue falls into, the family Flaviviridae, and this is the genus Flavivirus, there's a lot of different non-human reservoirs of flaviviruses. And importantly, there's a group of insect-only or mosquito-only viruses shown here that are actually sister to all of the other vector-borne diseases and a few that are non-vector-borne, or there's no known vector. And so this started us thinking about how are these kinds of viruses distributed in nature, and how are they related to the kinds of viruses that emerged to become human or non-human animal pathogens. So we now started to look inside mosquitoes to see if we can draw a synthesis about all the kinds of viruses that might occur in mosquitoes and how are they related to human vector-borne pathogens. So we sampled mosquitoes from different kinds of environments. In this case, we sampled mosquitoes from a range of habitats in a dengue endemic and malaria endemic and other pathogen, Japanese encephalitis endemic area in Thailand. So we sampled across a very large transect in southeastern Thailand using a variety of trap types to optimize the diversity of the mosquitoes that we could bring into the trap. We didn't want to just be biased towards human biting or anthropophilic mosquitoes. And we sampled the viruses from these very different habitats. Some of them were quite untouched by human efforts, and others were highly agriculturalized, and then some were urban or peri-urban environments. And the mosquitoes, in turn, were very diverse, and that diversity depended on the habitat from which we collected them. So they varied from mainly non-human biting mosquitoes to highly invasive, like Aedes aegypti and Helix fasciatus mosquitoes. They really specialized on feeding on humans or human or animals that were also affiliated with humans. This is a picture of a couple of my students and I sampling at a rice swamp. We used a variety, that was a light trap, this is a grabbing trap. And so then we take this mosquito. So this mosquito was feeding on me. I was a model, and she fed so much that she passed out. So I like to think of her legs thrown over her shoulder, like she just ate a big turkey dinner. She did not die. She actually revived. So she really fainted through overconsumption of blood. And then we popped her into a tube. So she was a photo model. Obviously, she's contaminated with my blood, and I don't have any pathogens. So we just used her as a specimen. But other mosquitoes are blood-filled, and they're like flying hypodermic needles sampling the environment of vertebrates they're feeding upon. And then we capture them in our tubes, and they have the signatures of not only their own mosquito-specific microbes, but the vertebrate-specific microbes and the vertebrate-specific genetic signatures that they house in their blood-filled cavities. So they're quite an interesting model. So when we take them back to the lab and we homogenize them, they don't survive that. And then we compare their microbial communities. And in this case, we did targeted next-gen sequencing of 2 amplicon regions of bacterial characterization, 16S, and a eukaryotic microbe characterization. And I just want to point out 2 things. We just started on the 3 main medically important species, Felix fasciatus, Aedes albopictus, and Aedes aegypti in this glimpse. And what we wanted to look at is just to see whether the characteristics of the microbial communities vary. You can see that on the outer edges of both of these images, these are the species are across the top, and the environments from urban environments are on the outside, and the rural environments are on the inside. And you can see there's, even within a species, there's quite a degree of variability by habitat. But in general, the species also have very unique microbial communities. Here I point out Wolbachia. Wolbachia is not known to infect Aedes aegypti, but it infects Aedes albopictus and Felix fasciatus. One of those Wolbachia was actually a Wolbachia of a dirofilarial nematode inside the mosquito. So we're getting a set of nesting Russian dolls of microbial associations, so it's really interesting. But the viruses that we pulled out of the mosquitoes do speak back to the origins of many vertebrate infectious diseases. This is a phylogram of the Buneviridae, and there are many viruses that infect humans. Hantavirus and its relatives here, Crimean Congo, Rift Valley fever shown here, and then we pulled out this virus, a quasi-caron-like virus, which is very closely related to a bunch of other insect-restricted viruses that look insect-restricted in cell culture and animal models. So this is just a blow-up of that clade, and you can see all these viruses here with Rift Valley in the middle, and the insect-specific viruses shown here, Posicaron, and some other insect-only viruses. So when we look deeper at the genome of Posicaron and compared it to Rift Valley fever and some other insect-only viruses, we saw that it was missing an important gene, a reverse encoded, a non-structural gene that ran backwards, was encoded in the reverse in vertebrate infecting viruses, and that gene was shown to be critical to infect vertebrates. So not only can we trace potential insect-only origins to vertebrate infecting pathogens, but we can look at the genetic mechanisms that enable them to make that emergence successful into vertebrates. So we looked at some other species of mosquitoes and found a similar pattern. Many of the viruses are specific to the host, and a few of them, even within a host though, are specific to habitats, some occurring only in rural environments and some, in urban and some in rural. So we're still preparing that work, but in general, we expanded some of our work to look at some California mosquitoes. Again, very diverse species, host specific in terms of their microbial and fungal communities, and also their virus communities. What was interesting is that we found viruses from many different families, and in all cases, the mosquito-only, there were mosquito-only lineages and up to 4 different virus families in this case. So we have the Buneviridae again, we have the Posicaron-like virus right here, and then we found 2 more viruses that were very unique to Helix pipiens that again suggest that there's very significant mosquito-only, mosquito-restricted lineages in these virus families that also spill over into humans. So again, in the narnoviruses, we find a group of viruses that are unique to Culex pipiens and that are basal or sister to a bunch of other viruses that have been characterized from different kinds of pathogenic associated. These are not in humans, but they're in yeast and in other organisms. Rhabdovirus, again, a unique mosquito-only lineage that's emerged from Culex pipiens and another Aedes syriensis, which is specific to California. So another one, double-stranded RNA viruses. So the picture that we're sort of developing here is that now we're still missing a lot of the biodiversity of RNA viruses in mosquitoes. And so the picture is still, we're not sure whether these viruses are sister, are basal, or simply developing as their own insect-restricted lineages. But we do observe that there are insect-restricted lineages across multiple major groups of viruses. And that many of these major groups of viruses do have pathogens of humans and other affiliated organisms. So we're suggesting, as we continue to characterize the microbial diversity of mosquitoes and their viruses, that there might be an important evolutionary sequence of moving from an arthropod-restricted lineage into an emerging vector-borne pathogen. So with that, I'll take any questions.